Hello, everyone. My name is Jocelyn Vincent, and I am a junior majoring in biochemistry and chemistry. I currently am working in Dr. Chowdhury's research group, and I will be discussing protein digestion using titanium dioxide. First, I will discuss why we perform protein digestion. Protein digestions are performed to break a protein into peptides that are easier to analyze. Proteases are used to digest proteins, and the most common protease is trypsin. Titanium dioxide can be found in sunscreens, cosmetics, and coating on medications. In addition to these applications, it has been successfully adopted for environmental purposes, such as wastewater treatment and air purification techniques. There are three known crystal structures of dioxide, which are anatase, rutile, and brookite. Figure 1 describes the anatase structure of titanium dioxide that we will be using for this study. It is known that titanium dioxide cleaves proteins through this free radical mechanism, which produces hydroxyl radicals, and is shown in Figure 2. Figure 3 shows a classical free radical mechanism, which results in the cleavage of the protein at the N-terminus containing proline. The workflow for this study involves a titanium dioxide-coated well plate and a 6-watt UV light at a wavelength of 365 nanometers, which is shown in Figure 4. A well plate is coated with titanium dioxide. Proteins are introduced into the titanium dioxide-coated well plate. Then a UV lamp is placed 1 centimeter over the well plate for 3 hours. The solution in the well plate is then extracted and placed in an Eppendorf tube and then centrifuged for 5 minutes. Then the supernatant is transferred into a fresh Eppendorf tube and prepared for LCMS analysis. We performed the experimental workflow on a standard protein, bovine serum albumin, BSA, and observed the results with tandem MS. This peptide sequence was confirmed with the B and Y ions, and as shown in Figure 5, this peptide contains a proline, which means that we have cleaved the protein at the N-terminus of proline. The disadvantages of using the protease trypsin for digestion is that it is time-consuming, and it is also relatively expensive. Another reason why we are exploring an alternative strategy is to cleave proteins is because basic conditions must be maintained in tryptic digestions. We are currently working to optimize the methodology of using titanium dioxide to effectively cleave proteins comparable to trypsin. We want to test other standard proteins with this methodology to understand the potential of titanium dioxide to facilitate protein cleavage. Lastly, I would like to thank the UROP program for providing me funds, as well as all of those in Dr. Chowdhury's research group that has helped me in this project. Thank you.